Hello, my name is Sergio Baranzini, and I'm a professor of neurology at the University of California in San Francisco. And I'm here to present our project that is part of the Proto-OKN ecosystem. Our project is entitled Connecting Biomedical Information on Earth and in Space via the Spoke Knowledge Graph. Our team is multidisciplinary and institutional. We have Peter Rose from UC San Diego, Sui Wang from the Institute for Systems Biology, Scooter Morris and Angela Risk-Jackson and Youngmei Shi are from UCSF, Sam Gebre, Amanda Saravia, and Einar Sawyer are from NASA, and Einar actually has a dual appointment. She's a physician. She directs the Center for Space Medicine at UCSF and has many collaborations with NASA. And Charlotte Nelson is our commercial partner from Made by Your Services. So what this project is about is to try to integrate multiple sources of information of data that we can gather from experiments done at the International Space Station and capitalizing on the very large amount of data that we gather at UCSF from millions and millions of patients into the knowledge graph Spoke so that we can integrate this and come up with predictions as to which types of conditions might be useful for us to recreate on Earth that could be used towards designing a more efficient, safer, and long-standing space travel. So, but let me start with the genesis of our project, and this has to do with how data can become information and how information can become knowledge, because this is important for our rationale of, first of all, creating a knowledge graph for biomedicine, and second, on how we can use it. So data itself is our facts, our results from a particular experiment. But data itself is devoid of any contextualization. Data itself is not organized, is not synthesized. However, data from multiple experiments can be organized, can be contextualized, can be synthesized, and this is when it becomes information. So information is contextualized data, is synthesized data, probably coming from multiple observations. And then information then can be connected. So the dots of information can be connected and knowledge can emerge from these connections. So knowledge is a higher order, if you wish, emergent property of a network of information. And this is very important because we think that particularly in biomedicine, there are an enormous corpora of individual data sets. Some of them have been converted into information, but the knowledge is not necessarily being capitalized from all the information available. So, and this is what we're trying to do in biomedicine. Part of creating a knowledge graph comes from the concept of trying to capture the complexity, the biological complexity, which is, by the way, unmatched by any other field. The complexity in biomedicine is such that it really requires some sort of contextualization in this way. If we visualize the hierarchical organization of biological complexity, one might start from the bottom up, where information trickles up the hierarchy. Usually one can start from information encoded in the genome or in the metagenome in this case. And then the next level up will be that of gene activity. So genes get transcribed into RNA molecules, and this is the most immediate phenotype could be interpreted. And then RNA molecules get translated into proteins. Proteins interact in sophisticated networks, and these networks are arranged in such a way that form pathways in which cells later on integrate and decide on an output. So a cell is a great integrator of information via multiple receptors and intracellular signaling pathways. A cell senses the environment and then decides whether to proliferate or whether to differentiate into some other cell or to die of apoptosis. So this is a very critical element in our organization. And then cells arrange into three-dimensional tissues and organs, and all the way up there we have the organism, the patient that doctors see. And the whole point of this is that there's been a paucity in integration across different layers, particularly because it is so rich, it's so complex, that people can spend their whole careers in one of these stages and not even bother about how this relates to the stage above or below, because this is such a complex undertaking. And this organization spans 4 orders of magnitude in space and 4 orders of magnitude in time. So this is just the metabolic map to get an idea. This is just one of those layers. How can you possibly follow the flow of information when you have this amount of complexity? So this is where the idea of the knowledge graph comes in handy. And we created Spoke more than 10 years ago now with an idea in mind that it will serve as a integrator of information coming from individuals, from patients that once doctors see in the hospital. And this is a high-level representation of what Spoke contains. Essentially, Spoke is a database of databases in a graph format. It contains more than 60 specialized databases and are connected via our know-how of biomedicine and our know-how in computational science, so that we can seamlessly query this graph and go across the different types of information. For example, one could go from a disease to a genetic variant, or from a disease to the symptoms, or from a disease to the organism, the microorganism that it causes it. And then we have compounds, which could be pharmacological compounds or metabolic compounds. These can be related to the protein targets of those pharmacological compounds, and so on. So we can navigate multiple layers in that organization in a way that is seamless to the user, and we can enable this via graph navigation or a graphical user interface that users can understand the neighborhood of a particular concept. So Spoke itself has been the engine behind an entire ecosystem of applications, which include collaborations with other universities, collaboration with research institutions, for example, to understand how microbiomes relate to health and disease. It has been utilized for drug discovery, of drug repurposing efforts. It has been used towards understanding genetics of diseases. And in this case, what we're utilizing Spoke is through a collaboration with NASA to create analogs of space flight, of the situations that astronauts may be experiencing during extended space flights, with information that we have from experiments conducted on the International Space Station from one end, but also creating analogs on Earth from millions of patients that are being seen at UCSF, from which we can generate spoke embeddings, as I will describe later. Also, spoke has been the engine behind a commercial application that is commercialized made by Services, which was a spin-off of this endeavor that was actually part of the Convergence Accelerator from the National Science Foundation in 2020 through 2022. So like I mentioned before, the concept of utilizing spoke is that we are interested in integrating information that comes from actual experiments performed in space and information from patients that we're gathering here on Earth. So how are we doing that? Well, the overall goals of our program is to create a layer of integration into NASA's Gene Lab. Gene Lab is a platform that now has been integrated into a much larger ecosystem called the Open Science Data Repository. And this incorporates hundreds of thousands of experiments that scientists from all around the world have been conducting via experiments at the International Space Station and at different research facilities on Earth, with simulating microgravity, simulating radiation conditions that astronauts might experience during extended space flight. So all of those experiments are now in the Gene Lab platform, and what we're doing with Spoke is enabling a new layer of integration across different data sets that before was not possible. The second objective of our project is to generate analogs of space flight utilizing a very extensive corpus of electronic health records from UCSF. More than 6 million patients have been de-identified. The information from these patients have been de-identified, and we can utilize that for research. And I'll show some examples of that. And then finally, our goal is to integrate these large efforts into the two phase 2 or stage 2 programs, FRINC and SPIDER, that will facilitate the fabric of the Proto-OKN connecting with the other 17 programs. So I would like to show at a very basic level how we're doing to integrate data from particular experiments in the Gene Lab database on the left-hand side with the Spoke knowledge graph on the right-hand side. So, for example, in Gene Lab, there might be a particular experiment that comes from a particular mission in which a particular condition, let's say mice were flown to the International Space Station, stay there for 30 days, and then they return to Earth. And the experiment was to understand the differences in gene expression in different organs that were stimulated by the 30 days in space. So with that particular experiment, we have many bits of information that we can embed onto Spoke. Most notably, what we did is we created an intermediate connector between the information in Gene Lab and Spoke so that we can embed onto Spoke the critical element of that particular experiment. So in this experiment that I'm talking about, mice were flown into the International Space Station. The gene expression profiles in liver, for example, were measured. So the result of the experiment is a long list of genes that are differentially expressed in liver in response to 30 days in space in mice. So what we do is we embedded that list of genes that were differentially expressed into Spoke, and then we diffused that information through the neighbors of Spoke in that, starting from the genes in the list. And that creates a complete version of a Spoke that now has weights. Each of the concepts in Spokes are weighted based on the initial list of genes that was embedded. And now this creates sort of a barcode of that particular experiment in the context of the entire network. What this allows us to do is that even though the input were list of genes, because Spoke connects genes to diseases and genes to proteins and proteins to drugs, now we can have a much more complete picture of what the organism might have been experiencing based on that input layer. And this comes from earlier work that we did in collaboration with NASA, that by embedding single gene expression experiments, for example, we were able to infer which symptoms those mice might be feeling during the space flight. So what we did is now we incorporated thousands of these experiments into the platform, and now any user with access to GeneLab can now run a Spoke analysis. So again, the code is open source, so any interested party can go to our GitHub page and include and can see how this this layer was programmed, so we can enable this additional type of analysis done on the OSDR platform. And we've also participated in an OHAX hackathon earlier this year, where participants of the hackathon analyzed particular data sets and used this platform for creating hypotheses and generating hypotheses about the results at a much higher level than it could have been obtained, for example, from single pathway analysis and so on. Currently, the types of data sets that are incorporated into this platform include transcription profiling by two different platforms, RNA sequencing and DNA microarrays, and also DNA methylation profiling by two platforms, by whole genome bisulfite sequencing and by reduced representation bisulfite sequencing. So these are two different platforms for DNA methylation profiling. And now we're working towards being able to incorporate microbiome information from 16S ribosomal RNA gene sequencing. And this is just a snapshot of the platform. If you go to the GeneLab visualization portal, you can select on the left, apply filters, and you can select the missions, the type of information that you're interested in, which type of organism, which type of tissues, and then you select an experiment and for any experiment that has information on gene expression or DNA methylation, you have the option to visualize them in Spoke. So you click this button, and then the Spoke analysis happens. It's been pre-computed, so it's really fast, and users can get a representation, network representation of the results. So let me move now to the second objective, which is generating analogs of space flight from the electronic health record. And for this, our train of thought is there are certain conditions on Earth that may resemble some of the conditions that astronauts endure during extended spaceflight. For example, astronauts may feel isolated during extended periods of spaceflight. They may feel lack of mobility. They will have incomplete access to fresh food. They will suffer from bone loss. So all of these are conditions that we can take advantage of to study in much, much larger numbers of individuals. The number of astronauts that have been to the International Space Stations is in the low hundreds. In contrast, we have access to millions of patients who might be, some of them, thousands or tens of thousands might be experiencing social isolation, lack of mobility, and so on. So the idea is to create cohorts of individuals, of those thousands of individuals with that particular condition, and then embed the information of those patients into Spoke, and then use those embeddings to extract information that might be useful for NASA to inform about spaceflight. So what are the conditions that could be reversed? For example, might there be any medications? If patients are taking a particular medication to counteract some of these conditions on Earth, maybe they can use that in space too. So this is the way we do it. I will leave the details of this for my colleague Peter Rose when he presents his master class on the technical aspect. But the idea is that we will generate these embeddings, as I showed a couple of slides ago, and create a propagated version of Spoke that is representative of the cohort of patients with that particular condition. In this case, social isolation or lack of access to fresh food, and so on. And so what again this will achieve is a set of variables that we will present to our collaborators in NASA to say, well, these might be actions or counteractions that could be taken to minimize the risk of bone loss or to minimize the risk of social isolations that might be felt by astronauts during extended spaceflight. And the last part of my introductory talk is about how we're planning to integrate with the projects Spider and Frink, which are the two horizontals in the Proto-OKN ecosystem. And in this case, what we're doing is we're interacting differently with both teams. With the Spider team, Spider uses a API sort of architecture that investigates automatically the architecture of our network, and they record that architecture so that they can do federated queries from users. The way that Frink works is Frink creates sort of an additional layer that they call it fabric, so that information from one graph can be connected to information in another graph. For example, we've collaborated closely with the BioBricks team, which is another Theme 1 group, in which they are interested in safety of chemicals. We have lots of chemicals, but they have all the safety information. So there's a great opportunity for cross-graph queries that that Frink and Spider can leverage if someone is interested in knowing more about which are the safety features of a particular drug, and then they want to learn more through Spoke what the drug might be doing, what might be the side effects when used in humans, what might be their therapeutic effects, what might be the molecular target of that drug. So all of these are enabled thanks to this fabric that the Frink team has developed that allows connectivity and federation across graphs. And the last part that I want to mention is we're actively exploring integration opportunities, and this is something that has become very useful, presented in the last few months, which is the utilization of the MCP servers. MCP stands for Model Context Protocol, and it works in such a way that it connects a large language model with any database. And the database could be a graph database, it could be a SQL database, it could understand Cypher queries, it can understand Sparkle queries. So the MCP client, when programmed appropriately, can allow prompts from a large language model and directly direct those and strictly direct those prompts to the appropriate database, extract the information from the database, and present the user with a response in plain English. And that response may include text, but also what it's called artifacts, which could be tables or graphical outputs, and even interactive graphs. So in this case, we're leveraging the MCP protocol for integration of not only our Spoke graph, but also other graphs. So this is a tool that we're helping the Frink team develop to leverage across the entirety of the Proto-OKN ecosystem. And some of the salient features of MCP is that first, it leverages a unified natural language interface. So right now, Anthropic Claude is the LLM of choice because it's the way that it is presented is the most effective and seamless to be integrated into the MCP protocol, but others may be ready to launch in the future, including ChatGPT. It features a modular server integration, which means that connects to multiple MCP servers, Sparkle or MySQL modules. So independent on how a particular knowledge graph is encoded, it could be in a property graph such as Spoke, or it could be an RDF such as BioBricks. It doesn't matter, it will connect both seamlessly. It provides a federated knowledge access. So it's a great tool for integration across graphs. It also features or enables automated knowledge discovery because it really facilitates and accelerates the access to information that otherwise it could take a long time to extract, and a lot of expertise in coding or in querying in those fairly technical languages. And finally, it acts as an interoperability hub. So it's the middleware that will integrate third-party resources with the Proto-OKN knowledge graph. So we're really excited about this. So in this case, we're working again, like I mentioned, with the Frink team to incorporate a Claude into the ecosystem of the Proto-OKN. and hopefully we'll be able to show a demo during the upcoming in-person meeting. So our project has a website. This is the URL, and in the website you can find more information about our progress, how to contact us, information about our reach out opportunities, and opportunities for collaboration. Please feel free to reach out to us. In terms of our stakeholders, we're partnering in this with NASA. NASA is super engaged in this collaboration. Two of our members have recently presented at the investigator workshop in Texas. We participated in NoHacks hackathons. Spoke is now part of an OSDR working group. So the OSDR team has multiple working groups in which investigators from all over the world can participate and present ideas and present challenges. And so Spoke for Space is now has its own working group right now. And also Made Bio continues to grow. There's a number of customers that have been inspired by the progress done by this team. And again, this is with support from the NSF Conversions Accelerator program that Made was spun off. So we're really happy to see it continues to grow. And with this, I would like to end my presentation, and thank you very much.